So then we're going to be talking about protein structure, function, and folding. Chapter 4. So I guess the learning goals are going to be the structure and properties of the peptide bond. And we talked about that a little last time. Like jumped ahead into this chapter. You guys remember anything about the structure and properties of a peptide bond? That amide bond? Huh? A week ago, yes. It's very important, this portion of this peptide bond is characteristic. Right? If you remember, there's like a barrier to rotation about it. You guys remember that? There's a resonance structure you can draw, so there's sort of like, it's, you know, those overlapping pi orbitals, because it's, you know, the resonance structure is halfway in between both, so you got those pi orbitals in that bond. So it's like it's almost a double bond, but not quite a double bond. It has double bond character, that peptide bond, so there's that restriction to rotation around that. You got to put energy into it to get it to rotate around the double bond. So normally, you don't see rotation around that peptide bond. And in the structural hierarchy of proteins, we're going to learn about structure and function of some fibrous proteins, and then structure analysis of globular proteins, and protein folding and denaturation. You guys know what denaturation is?
Unfolding the protein?
Yeah, exactly. Just unfolding the protein is denaturation. So, do you guys know of any examples of fibrous proteins? I think I talked about some last time based on just certain amino acids that are modified. But I doubt you remember that one. That one's not as important as the peptide bond. Any ideas? Can you think of any fibrous proteins?
Actin?
Yeah, there's actin fibers, but really actin fibers are polymers of monomers, actin monomers. And when you talk about the monomer of actin, that's normally called G-actin. Do you know what the G stands for? Globular. Right, so actin itself is a globular protein. Normally structural proteins, and actin is a motor protein also, and that's where you have the fibril actin, which is a fiber of globular actin. So it's not necessarily a fibrous protein, although they do refer to it as F-actin when it's a polymerized like that. Although it's not technically polymerized where there's not covalent bonds holding it together. It's just held together via non-covalent forces. So, yeah, fibrous proteins, and I was doing this, looking at my nail. Do you know what protein makes up your nails? Keratin is a fibrous protein. Collagen is a fibrous protein. And then there's the two keratins, beta keratin and alpha keratin. And the beta and alpha kind of help describe the main secondary structure of those proteins. We'll look at that a little bit also. So, structure of proteins, unlike most organic polymers, protein molecules adopt a specific three-dimensional conformation. And what governs how it's going to fold into a three-dimensional structure?
Intermolecular forces and hydrophobic interactions.
Yeah, those are all like interactions, but what specifically about protein?
Sequence?
The sequence, yeah, the amino acid sequence. But yeah, the way the intermolecular, well, I guess there's some it's involving the solvent, water, or the intramolecular forces within the protein itself.
Yeah.
But yeah, that sequence causes that to have the native fold, and which gives it that specific biological function. The native fold has a large number of favorable interactions within the protein. So these are energetic, favorable reactions. And there's a cost in conformational entropy, right? When it's folded, it's not as free to rotate around like bonds, and the entropy is lower when you have that folded. But you get entropy gains, and the main entropy gain you get is from what, you know? The solvent, water. So you bury all those hydrophobic R groups, and then that water, the solvent, doesn't have to be highly ordered around those hydrophobic R groups. Buried it. That's why a lot of proteins, they're globular, like we just saw on the last slide, globular proteins. They get that name because they're like little globs of oil kind of thing. Like they're spherically shaped for the most part. So, favorable interactions in proteins are going to be this hydrophobic effect, which is what I just was talking about. That's where you get the most free energy from a protein folding comes from that hydrophobic effect, the favorable entropy you get from the solvent portion of that folding. Hydrogen bonds, you get a lot of hydrogen bonding in protein structure. We're going to see the secondary structure is all hydrogen bonding between the main and mid molecules in the bond. You know, the carbonyl and that nitrogen, they form hydrogen bonds between each other, and that's one of the favorable main structures of alpha helices and beta sheets, which is secondary structure. London dispersion, so we get medium range, weak attraction between all atoms contributes significantly to the stability of the interior of the protein. You'll get a lot of that. The aromatic amino acids, anybody remember what those were called? An example of an aromatic amino acid. Do you know? Proline is not aromatic.
No, but it's other.
It's a ring, but it's a big difference between it being aromatic and phenylalanine is probably the easiest one to remember, right? Because it's a phenyl group. And then there's two others, tryptophan and tyrosine. So, and then electrostatic interactions. You'll see proteins will form salt bridges between two charge groups to help with their three-dimensional structure. So there's 4 levels of protein structure. Have you guys seen this before? Yeah, so this is the basic stuff. Primary structure is the amino acid sequence. Secondary structure is going to be where it forms these localized structures such as alpha helices or beta sheets. That would be considered secondary structure. Tertiary structure is the overall fold of one polypeptide chain. And we know a lot of proteins don't function as one polypeptide chain. In this example, anybody know what molecule this is? Hemoglobin, yeah. Hemoglobin exists as 4, polypeptides together to gain the function. And this is, as we all know, it's used to carry oxygen through our blood. And that, the way it does that, the way it's able to release oxygen in the periphery and gain as much oxygen as possible in the lungs has to do with having 4, different four pieces of it together. It works together and it changes its affinity for oxygen depending on the pH and the So structure of the peptide bond, so we talked about this a little last time, it's partially dictated by the properties of the peptide bond. The peptide bond is a resonance hybrid. Didn't I make you guys draw that, the resonance structure last time? And the resonance causes those peptides to be less reactive compared to other esters and quite rigid and nearly planar. So there's a barrier to rotation around that peptide bond due to this resonance hybrid. And it also exhibits a large dipole moment. Through those, when we draw that resonance structure, you have those charge groups. So the reality is there's partially charged in the reality of that structure, right? In the one resonance group, there's no charge, but the other, you have a direct charge on the nitrogen and oxygen. So the reality is a semi-charge on that nitrogen and a semi-negative charge on the oxygen. And this, I like this figure. This one's not in your book, it's one of the older books. But this shows you that the sharing you get of those pi electrons throughout the peptide bond. So you have a plane, and these pi electrons are all shared, so you have like barrier to rotation around this carbon and nitrogen bond that is the peptide bond. So, but there are some other groups that aren't quite as rigid. So you'll have a group coming off this alpha carbon, and then this, between this carbonyl and the alpha carbon, and this and the nitrogen, those are able to rotate. And we give those a certain name. So you do get rotation. The phi angle, which is the angle around the alpha carbon and the amide nitrogen, which is alpha carbon and an amide nitrogen, that's your phi angle. And then your other angle is the psi, and that's the angle around the alpha carbon and a carbonyl carbon bond. I keep going wrong. Well, I guess it shows it both. But this is the alpha carbon and then a carbonyl carbon that's involved in the peptide bond. So this would be your psi angle, the angle between that, and this would be your phi angle. I think you can see it a little better. I don't know why I was going back. I could just come forward. Because you can see it a little better in this figure that your book has. So this is a phi angle, this is around this angle, and this is your, well, did I say that wrong? I don't know my Greek letters. It's never in a frat or anything. Phi and psi. Right, is this phi and that's some? Or is that phi and that's some?
Phi is the one with the circle. Phi is the circle.
Phi is the circle? Yeah. So that's the one that's involved with the amide nitrogen and the alpha carbon. Psi is involved with the carbonyl and the alpha carbon. So distributions of phi and psi dihedral angles. Some phi and psi combinations are very unfavorable. Right? So what might be unfavorable is if this is like, there's a better picture, but not this slide. But you're going to have some that aren't favorable at all because you're going to have steric interactions between the hydrogen off the amide or the R groups, right? The R groups will interact also. And those aren't going to be favorable, so you get steric crowding. Some phi and psi combinations are more favorable because of the chance to form favorable hydrogen bonding interactions between those amide hydrogens and the carbonyl oxygen in the backbone. And those favorable, if you plot phi and psi angle distribution through a peptide, you'll see certain ones you always see over and over again, and they're associated with the type of secondary structure that is being formed. So you have parallel beta sheet structures are always found around this here. Anti-parallel beta sheets are right here. You guys have no idea what the parallel or anti-parallel means just yet, right? Huh? Okay. And then you're going to see right-handed alpha helix here, and then a left-handed alpha helix here. And then there's also a place, a collagen triple helix. So collagen, we've learned, was a fibrous protein. It's a structural protein that has a special type of helix that's not an alpha helix that gives you its own special phi and psi angles that you'll see on a Ramachandran plot. I don't know if I say that correctly. Ramachandran. This is what it is, Ramachandran plot. Plotting the phi, or this is phi, right, and psi angles. And then this shows you a Ramachandran plot. And all the amino acids, you have the plots of their phi and psi angles. Now we'll talk about secondary structures because this kind of plots out where you might find certain secondary structures. So secondary structures refers to the local spatial arrangement of the polypeptide backbone. And this is going to include your alpha helices and beta sheet structures. And you got to remember the alpha helix is stabilized by hydrogen bonds between nearby residues. Okay? And these are hydrogen bonds of the peptide backbone themselves. A lot of people confuse, because you can have interactions between the R groups, and some of those R groups can be hydrogen bond donators or acceptors, right? But for secondary structure, the hydrogen bonds that stabilize the secondary structure are always between the amide portions of that peptide, that carbonyl and the nitrogen that's involved in the peptide bond themselves. Okay. Secondary, for alpha helix, these hydrogen bonds take place between nearby residues. And beta sheets, they can be stabilized by hydrogen bonds between adjacent segments that may not be nearby. They could be totally far away in sequence, but they happen to be close enough together in spatial arrangement where you're able to stabilize this type of secondary structure, from their hydrogen bonds. And then you can also have irregular arrangements of a peptide change, and those are called random coils. They're not necessarily coiled, they just call them random coils just because they can exist in these random structures. A lot of times they're just unstructured. Any unstructured region that doesn't have regular structures, often called a random coil. Okay. And this shows you your idealized bind side angles, secondary structures. Don't memorize these numbers because I'm not going to quiz you on numbers like this. I'll tell you about the numbers. I'll quiz you. I'm going to quiz you on numbers that you have to calculate. So the alpha helix, helical backbones held together by hydrogen bonds between the backbone amides and the N plus 4 amino acids. So you have a right-handed helix, typically has 3.6 residues, it's 5.4 angstroms per turn, and peptide bonds are aligned roughly parallel within the helical axis. Side chains point out away from the helix. So here you could have a left-handed helix, you can tell if it's left or right. So the sequence goes from the N to C in an upward direction, right? So the amino terminus, carboxy terminus would be up here, and depending on how it's turned, it's like if it's a right hand, it's going like this with the C terminus up top, left hand like this. So they always mention this in biochem. I don't know when you'd ever use that. Unless you're doing like crystallography, where you're actually mapping this stuff out or analog. So the inner diameter of the helix, no side chain, is about 4 to 5 angstroms. Too small for anything to fit inside it per se, but the outer diameter of the helix with the side chains is about 10 to 12 angstroms, which happens to fit quite nicely right inside the major groove of a DNA molecule. So hence, you'll probably, DNA binding protein's probably going to have an alpha helix domain where it's binding to the DNA. What kind of sequence gives an alpha helix with one hydrophobic base? Do you know what this means? What is this question asking? So, if we looked at the alpha helix, R groups are going to project out of this helix away. So, if you're going to have one face, one side of this face is going to be hydrophobic, the other hydrophilic, you're going to have not necessarily alternating sequences of, hydrophobic amino acids and hydrophilic, but close to alternating. Because how many per turn is it? I think 3 point something per turn. So, and this is called an amphipathic helix. You guys heard of that? I think we talked about amphipathic molecules. This would be an amphipathic helix where you have a mix of hydrophobic and hydrophilic amino acids, where hydrophobics are all on one side and hydrophilics are on the other. Typically the hydrophobic helix, depending on what type of protein it is, will either be facing towards the inside of a globular protein or towards the inside of a membrane or a transmembrane helix. The hydrophilic on the inside of the membrane transporter or the outside exposed to the solvent water for a globular protein. And this is, as you can see here, this is a picture of an amphipathic helix, but an alpha helix. And the R groups are always projected out away from the helix. And if you look straight down, projecting away from the helix, and these are just different models. This shows you an amphipathic helix. You can imagine here, like here, they're showing you the blue is a cationic amino acid, the red is an anionic, the yellows are hydrophobic, so we have hydrophilic charged, hydrophobic, hydrophilic. This could be, I don't know if they specify what the grades are, but hydrophilic, that could probably be hydrophobic, depending on if it's buried inside of the protein, globular protein. Sequences affect helix stability. So not all polypeptide sequences adopt an alpha helical structure. Small hydrophobic residues such as alanine and leucine are strong helix formers. And then proline acts as a helix breaker. Can anybody tell you why? Why proline might be a helix breaker? You said something about proline earlier.
Yeah, it's got that weird bond between the nitrogen.
Yeah, so it's not necessarily a true amino acid. It's an amino, like the I amino acid, because it's that cyclic structure. So it's limited. Rotation around the amide and the alpha carbon are almost impossible because of that cyclic structure of the proline. So it can only take a certain, is that 5 or 5? Don't remember these things either. So it's only going to have a certain angle, so it breaks. You'll see prolines a lot of times where the alpha helix is going to stop, go to a random coil, or actually in the lipoproteins, you'll see a proline multiple times where you have a helix, proline helix, and it kind of bends around your globule of fat that the lipoproteins carry around in the bloodstream. And then this is a table from your book showing you the propensity of amino acid residues to take up the alpha helical formation. And they have delta G. I don't know, I forget this stuff. About this, but it just shows you some things are highly likely to form an alpha helix. Proline obviously is not, it's got a greater than 4 delta G. And remember alanine and some of the others were likely to, obviously glycine is not, you'll often see glycines at turns and whatnot also. Anybody know why glycine you might see at a turn or a bend? It's the smallest amino acid, so it's only got a hydrogen for the R group, so it's not going to sterically inhibit like a tight turn. You'll see that also. One of the things, remember, about the peptide bond is the partial charge. And that'll cause these helixes to have like a net dipole type interaction. If you recall, the peptide bond is a strong dipole, the carbonyl slightly negative, the amide is slightly positive, so if you have a helix, those stack up on each other, gives the net helix, makes it like one large dipole itself. And negatively charged residues often occur near the positive end of the helix dipole, so that makes sense, kind of counteracts that net dipole. And here, it's like a graphical representation, you can see the net dipole. You have negative dipole here with some positively charged residues. Negatively charged residues around the positively charged dipole interaction on the helix. Now we'll jump to beta sheets. So the planarity of the peptide bond and tetrahedral geometry of the alpha carbon create pleated sheet-like structures. So this is what you'll see when you don't see alpha helices, you often see beta pleated sheets. And it kind of looks like how the toilet paper or whatever kind of fold out is like, or like a pleated sheet, or like those old fans that we used to use to cool ourselves. It's kind of like that pleated structure. Or these are the peptide bonds, planar like this. And if you notice on the pleated sheets, the R groups, they project up, back and forth, up and out, up and down, rotate back and forth, how they're going to project away from the pleated sheet at 90 degrees. So, like I just said, side chains protrude from the sheet, alternating in an up and down direction. So oftentimes, if you see a pleated sheet at the interface or in the outside of a globular protein, our groups projecting inside will have a hydrophobic domain. Our groups projecting outside will have to be hydrophilic. They will either hydrogen bond or charged or whatnot, so they can interact with the water. So parallel and anti-parallel beta sheets. What do we mean by this? It's because these beta sheets, the hydrogen bonding interactions are here between the oxygen and the nitrogen. So you have to get sheets close enough together to be able to hydrogen bond and stabilize these beta sheets. And whether they're parallel or anti-parallel, it's the direction of the chains. So parallel beta sheets are where the direction go the same direction, from the amino terminus to the carboxy terminus. Anti-parallel is where one goes from the amino terminus to the carboxy terminus, the other goes from the amino terminus, the opposite direction to the carboxy terminus. And the parallel beta sheets, the hydrogen bond strands run in the same direction, and this results in a bent hydrogen bond, right? The bent hydrogen bonds aren't as high in energy as the direct straight hydrogen bonds that we see in anti-parallel sheets. So it's, you have the stronger hydrogen bond with anti-parallel, weaker hydrogen bond with the parallel. This next figure shows you those hydrogen bonds in an anti-parallel sheet. So here we have an anti-parallel. This is the amino terminus to the carboxy terminus, and vice versa, going the opposite direction. So those strands are going opposite, so they're anti-parallel beta sheets. So a lot of times you might see an anti-parallel beta sheet where it's real close in sequence, because you could have just a beta turn here with a glycine or two, and then continue the sequence here. Or this can go off, have some alpha helices, some more over here, and then come back. So it could be a long distance in the amino acid primary sequence away from each other. They could be fairly close to the anti-parallel. Parallel beta sheets, though, are like this, so they're both going amino terminus to carboxy terminus throughout, and they go the same direction in all the corresponding. If you notice, they're not exactly straight with the hydrogen bonds, so they're not as strong either. So, and these are often far apart in amino acid sequence, because you're going to have to get some. You go from here, it's going to have something in sequence that's going to connect it eventually to this portion. Not only that, but sometimes you can get parallel or anti-parallel beta sheets that form between two proteins where you have binding partners. One protein is a binding partner to the other. They may form beta sheets together to form that bonding interaction. We talked about turns. They're referred to as beta turns. Beta turns occur frequently whenever strands and beta sheets change in the direction. The 180-degree turn is accomplished over 4 amino acids. So it takes 4 amino acids to do that 180-degree turn. And it's stabilized by hydrogen bonds from the carbonyl oxygen to the amide proton, 3 residues down in sequence. Often we'll see a proline in position 2 or a glycine in position 3 are common in these beta turns. And this shows you the type 1 beta turn. And here, 4 amino acids. Here you have the carbonyl oxygen, hydrogen bonding, the amide, hydrogen in from amino acid 1 to 4. Here you have a proline in position 2. In position 3, oftentimes in the type 2 beta turn, you'll see type a glycine in position 3. Position 3 of the type 2 beta. My words keep jumping. I warned you. Prolines themselves they can be isomers, and there's actually enzymes that can change the isomerization of prolines from cis and trans. So for peptide bonds involving prolines, about 6% are in the cis configuration, and most of these 6 involve beta turns. Isomerization is catalyzed by enzymes known as proline isomerases. Anytime you see an ACEs behind something, typically it's some sort of enzyme. Normally you can decipher what the enzyme catalyzes, what reaction that's going to catalyze. obviously, this proline isomerase. I can't say that. Can you say that? Yes. See, I should be able to say that because I said it a billion times before, but not today. So proline isomerases are going to catalyze the isomerization of proline, right? From cis to trans, and this just shows you the cis and trans. So trans, that ring's poking out. Cis, that ring's poking in, and if you look at cis, it makes it look like a good beta term. Right? So the protein tertiary structure, what was tertiary structure? Huh?
Third level.
Yes, the third level, so that's just the one polypeptide chain. So you're going to have secondary structures interacting together to form that tertiary structure. So it's the overall spatial arrangement of all the atoms in one protein or polypeptide. Stabilized by numerous weak interactions between amino acid side chains, largely the hydrophobic and polar interactions. Hydrophobic are going to cause, those hydrophobic regions to be buried away from water, the solvent, and they can also be stabilized by disulfide bonds. What amino acid can form disulfide bonds? Do you guys remember? There's only 2 amino acids that can't contain sulfur. Do you remember those? Methionine is one that contains sulfur. Do you think methionine can form a disulfide bond? Look at the sulfur on that methionine. Huh?
It doesn't have to.
It's got a methyl group on the sulfur, so that one's not involved in disulfide bonds. But the one that is should be on here too. So we've got a thiol group, or an SH. So you can form a disulfide bonds between 2 cysteines, right? If those cysteines are oxidized, you form a disulfide bond. If they're reduced, you add the hydrogen back to them, and they're just 2 free cysteines. What were we talking about? Can be stabilized by disulfide bonds. I mean, the interacting amino acids are not necessarily next to each other in the primary sequence. I already talked about that with beta sheets, but it can also be with the side chains interacting with buried side chains. They don't necessarily need to be next to each other in the protein sequence because you could have all kinds of alpha helix or loops or beta sheets bringing them together. And there's 2 major classes of proteins of tertiary structure, fibrous and globular. Fibrous are often involved in structure, so they're not too soluble in water, whereas globular are often soluble in water or lipid soluble if they're buried in the lipid membrane. They're kind of reverse like cells at that point, right? In globular, we see the majority of enzymes are going to be globular proteins. Structural proteins are fibrous. And this is the structure of collagen. So if you look, it's got this helix-like structure. Each chain of collagen has a helix, but the overall structure of collagen, you have 3 helixes together, what forms the collagen triple helix. You have 3 chains. That's what this figure shows you, 3 chains all together. And this triple helix is stabilized by hydrogen bonds between the R groups. If you guys remember, we talked about modified amino acids a little bit last time. Remember what amino acid, one of the amino acids gets modified a lot? And collagen anyways, would be proline. It gets hydroxylated. After it gets incorporated into the sequence, the proline will get hydroxylated, which adds hydroxyl groups or alcoholic groups to that proline. And those hydroxyl groups are involved in the stabilization of this triple helix between the R groups. And you get hydroxylysine also. So collagen, you'll find with most fibrous proteins, they're repeats of the same amino acid sequence over and over and over again. Collagen is mostly proline, glycine, and glycine. It's mostly proline and glycine for the most part. And this is just an example of a fibrous protein. And I don't know if you can tell, this is the 3 different helices looking down that collagen triple helix, or like staring down. And this is an example of a globular protein, and you can see it kind of has that oil droplet shape almost. This is your ribbon diagram. These are all alpha helices. This looks like myoglobin, or one of the subunits of hemoglobin. This is going to be your heme structure here, and this is the space filling model. So you see all those side chains are buried. So fibrous proteins from structure to function. So you can have alpha helix cross-linked by disulfide bonds, and they're tough, insoluble, protective structures. And this is what we find in alpha keratin. So alpha keratin has a lot of alpha helices that make up that structure, and it makes up hair, feathers, and nails. Beta conformation keratin, you have soft, flexible filaments, and that's what we see in beta keratin, or silk fibrin is made from beta keratin. So it's beta conformation, a lot of beta, cross beta sheets. And then the collagen triple helix, you have high tensile strength without stretch. You find collagen in tendons and bone matrix. So this is the structure of alpha keratin in hair. So you have alpha helices, typically consist of 7 residue repeats where you have A, B, C, D, E, F, and G repeated n number of times. So it's just over and over again the same sequence, where A and D are nonpolar, which is going to promote association between helix, where you have nonpolar helices and nonpolar helices. Remember, the R groups project which way from alpha helices? Yeah, outward, away from the helix. And these are going to be amphipathic helixes, right? If you have the nonpolar on A and D, right? So that's about every 4 amino acids, so they're going to be on the same side of that helix. The nonpolars are going to be sticking out, and they'll be able to interact with other nonpolar residues sticking out of other helices, and then which promotes this 2-chain coil to form between the alpha helices. Many of those can come together to form a protofilament, and then those protofilaments will come together and have this large coil that looks something like this that form in the hair. It's all alpha helices that form that alpha keratin. I'll get the same picture twice. Not yet, the next. And this is the structure of collagen. So collagen is important in connective tissue, tendons, cartilage, bones, cornea of the eye. And each collagen chain is long, glycine and proline rich. Like I said, glycine and proline rich, left-handed helix. 3 collagen chains intertwined into a right-handed superhelical triple helix. So collagen has that triple helix. High tensile strength, then a steel wire of equal cross-section. So super strong, and that triple helix will assemble to a large collagen fibril. Remember, it's those modified proline hydroxyl groups and whatnot where you get that strong hydrogen bonding between the helices. And this, we saw this, your single collagen helix and then your triple helix. And then those triple helices can come together, or you get multiple triple helices intertwined, makes a very strong collagen molecule. And the forces, you get this 4-hydroxyproline in collagen, and you've got to remember that this is modified once it's incorporated into that peptide sequence. It's not modified and then incorporated. Proline's incorporated into the peptide sequence via the ribosomes, and then it gets modified. And gives that, the forces the proline ring into a favorable pucker, offers more hydrogen bonds between the 3 strands from that hydroxy group. And post-translational processing, this is what I was talking about. That hydroxyl group gets added after the mRNA is translated into the peptide. And vitamin C or ascorbic acid is a critical cofactor for that reaction. Prolyl hydroxylase is the enzyme that does that. It requires alpha ketoglutarate. Anybody know what kind of amino acid that is? What kind of charge would alpha ketoglutarate normally have? What kind of charge does acetate or citrate have? So anytime you see like an 8, it's the charged version of an organic acid, right? So an organic acid, when it loses its proton, has what kind of charge? Negative charge, right? So anytime you see the alpha ketoglutarate, this is the charged version of alpha glutamic acid, or glutamic acid rather. And molecular oxygen and ascorbic are all required for that, adding the hydroxyl. So, and this shows you the interaction of hydrogen bonding between the triple helix. So you can see that. This is the overall reaction. So you have proline, alpha ketoglutarate. Actually, it's not an amino acid yet, right? This is the alpha keto of an amino precursor of amino acid. Oxygen and iron. Add the oxygen here, and this gets reduced to succinate, to carbon dioxide, and then alpha ketoglutarate and isocorbate with oxygen can then generate succinate and dihydroascorbate. Your silk fibrions is the main protein silk for moths and spiders. And you know spider silk is super strong. It gets that strength from its structure, which is an anti-parallel beta sheet structure. So you can think of this as the beta keratin. Small side chains of alanine and glycine allow the close packing of sheets, and the structure is stabilized by the hydrogen bonding within the sheets, and then London dispersion interactions between the sheets. So you have small side chains of alanine and glycine. And if you remember, what about the sequence of beta sheets? How do their side chains project? Up and down. So you'll have a lot of, in the sequence of fibrion, you'll have a lot of repeating alanine, glycine, alanine, glycine, alanine, glycine, right? Which is what this figure shows, where you have the glycine side chains all on the same side on interacting sheets, and then the alanine side chains will interact together on interacting sheets. And it's these London dispersion forces interacting gives you that strength and flexibility of the fibrion or beta keratin structure. We all know spider silk's used for webs, eggs, sacs, and wrapping their prey. Extremely strong material, stronger than steel, can stretch a lot before breaking. And people are trying to use it as like a composite material too. Crystalline parts, fibrin-rich, or rubber-like stretchy parts. Anybody have brown widows at their house? They're all over around here. They make those egg sacs with little spikes. Those things are tough as nails, and try to clean those out. They're like impossible to get out. So that's it for the structural type proteins. Now we're going to talk about motifs, which are secondary structures or combinations of secondary structures that we see a lot of in different proteins. So they're specific arrangements of several secondary structural elements. For example, we have all alpha helices or all beta sheet or both, and the motifs can be found as recurring structures in proteins throughout different proteins. And proteins are made of different motifs folded together often. One example is this beta alpha beta loop, where you have, what kind of beta sheets are these? Parallel, yeah. So these are parallel beta sheets, and you need something to connect them, which gives you that alpha helices, right? So the alpha helices here, you have the amino terminus down here, beta sheet runs this way, and then the alpha helix connects the two parallel beta sheets, and that's what's known as a beta-alpha-beta loop.
So that's a right hand loop.
So this is going to be, yeah, right. Well, wait, no. Is that right? Yeah, that's right. It kind of curls this way. Right hand loop. You can also have typical connections in all beta motifs. So this is an all beta motif. In your typical connection, you're going to have, it looks staggered, right? So you have an anti-parallel where it just loops and loops and loops and loops. Rarely you're going to see crossover type of connections like this. And then you have right-handed connections between beta strands. These are parallel. Left-handed connections are going to be more rare. And then you can have these twisted beta sheets often. You'll see those in like beta barrels. Like pouring, I think, is what you use to the kidneys to pull water into the kidney. Have like this pouring structure that's all twisted beta sheet structure. Beta alpha beta loop is where you have parallel betas. It's beta, and then alpha helix, and then a beta. And then you can also have an alpha beta barrel where it's a combination of beta sheets and then alpha helixes surrounding that beta sheet. So these are what's known as motifs that you're going to find in amino acids. Protein structures throughout repeated different proteins also. Quaternary structure is formed when you have multiple individual polypeptides associated together that form a larger functional protein. Hemoglobin is just an example of something that has quaternary structure. I do believe this is hemoglobin. Four peptides form together and form the overall hemoglobin structure. So how do we figure out these structures? Is some of the steps you're able to figure out protein structure. One of the main ones is X-ray crystallography. The problem with X-ray crystallography, some of the things you have to do, first you have to purify the protein. It has to be super, super pure so you get a nice clean crystal form. Crystallize the protein, and then you hit it with X-rays and collect the data on how those X-rays are diffracted from the crystal. You can calculate the electron density from that diffraction data and fit different residues to the data to come up with a three-dimensional model of how that protein structure looks. The pros for that is there's no size limits. It's well established. This is how they initially developed a lot of the protein structure that they discovered is through X-ray crystallography. It's really difficult for membrane proteins to crystallize this way, and you cannot see the hydrogens in the crystallization there. It's just little protons, right? So you don't get that. However, this shows you some pictures of the crystallization. So this is what the diffraction data looks like, and then you can piece that together to show you the overall alpha helices.
Are those computers?
Yeah, those are computers. So you take the diffraction data, feed it to a computer, and the computer spits this out. Yes, these were the old days before computers. You'd spend months doing the calculations by slide rule, right? I think they had computers when they started doing this. Another way, which I actually worked in a lab that did a lot of this, was NMR, structural methods using NMR. You guys look at NMR in organic chemistry. Nuclear magnetic resonance. So again, you have to purify the protein, and you have to dissolve that protein in water. Or what we used to do in my lab is they did just specifically looking at the transmembrane domains of nicotinic receptors, and they would just express that transmembrane domain in bacteria, purify it from the bacteria, dissolve it, but it's not soluble in water, so they would dissolve it in micelles. Do you remember what micelles were? They're like detergents. You have one big like hydrophobic domain and then a charged domain, and they form like little globules, and you're able to dissolve the transmembrane domain of the protein in the micelle, and still the micelle is small enough where you can get the spin needed to do the NMR. So, you know how NMR works, you hit it with that magnetic field, it aligns all the molecules based on the protons according to the spin, and then you take it away, and as those drop back down to their normal state, you release that energy, and it's going to be dependent on, you know, the environment of those nuclei, right? And it could be protein NMR, C13. There's a number of different nuclei that you can look at with NMR. So, but the pros is you don't need to crystallize the protein. That was one of the long, hard steps that would take practice to get a good crystal of the protein to be able to do the crystallography. And you can also see the hydrogens. If you're doing proton NMR, you're actually using the hydrogens to determine the structure itself. The cons, it's difficult for insoluble proteins. I just showed you one method to do the insoluble. And it works best with small proteins, right? Because they need to be able to have, you know, spin. And larger proteins are going to spin slower. Just, you know, it's the nature of Brownian motion, right? The larger things are going to spin slower, the smaller things will spin faster. So it works better with small proteins. And this shows you what the NMR data looks like. So you do this is a two-dimensional, looking at different proton shifts. You can do three-dimensional, where you look at proton chemical shift, C13, and I forget which nitrogen isotope was good. Do you guys remember? Probably haven't heard. Huh?
Never heard of nitrogen NMR.
Yeah. I think it's just the regular nitrogen. It's good. But I don't remember. 15. So, yeah, nitrogen, you could do three-dimensional spectra and put it together and determine where things are. Not only can you do that, but you can like verify your data by mutating, or by not mutating, but growing these peptides, because you got to purify these proteins. Often they're grown in recombinant systems. You can grow them with all C13 carbon available, so they're all the C13. Normally when you do regular proton NMR, you can do C13 because a small fraction of the carbons there are going to be C13. It's just a tiny fraction. If you're growing a protein, you can grow it with media that's nearly 100% C13 amino acids supplemented. So all the amino acids incorporated will have the C13 or the N15, vice versa. Or you can include certain amino acids that you don't want to see if you take away that proton spectra. Like you can put deuterium instead of hydrogen in places to see how it changes the spectra, see where that particular amino acid is. So, intrinsically disordered structures, these were called random coils, right? So some contain protein segments of that lack definable structure, and we refer to this as random coil, composed of amino acids whose higher concentration forces less defined structures. So lysine, arginine, glutamic acid, glutamate, and proline. Disordered regions can conform to many different proteins, facilitating interaction with numerous different protein partners. So if you have a lot of lysines together on a chain, it's not going to want to readily form like alpha helices or beta sheets, because you have all the same positive charge. If you guys remember, lysines are positively charged. Those positively charges are going to be repelled, right? So it's not going to, they're not going to want to be together. So it'll cause it to be random. Now you can change the pH from that lysine. If you guys remember, the pKa of the side group is up around 10.5 or so, or 10, I forget. So if you take the pH of a polylysine chain, it'll be random coil up until you get that pH up to about 11 or 11.5, about 1 pH unit above the pKa of the lysine. So then that's going to take the charge away from the lysine to now form alpha helices all day long. And this is circular dichromacy. the Ponder score, which is this is throughout protein. You take all the residues from the first residue to the last, and you plot it, whether, what it's going to form. If it readily forms alpha helices, it's going to be structured or non-structured, and you can determine which areas of the protein are going to be random. And in this case, you see the C-terminus has a random arrangement, and the N-terminus. And then this C-terminus happens to bind to random arrangements of these other groups here also.
What's Ponder?
What is Ponder? Predictor of natural disordered regions. So the higher the Ponder score, the more likely you're going to have to see here it's a very high Ponder score. So when we ponder a lot, our thoughts are more disordered. I can't sleep at night, just thinking randomly. So proteostasis, what is that? So that's the maintenance of our cellular protein activities accomplished by the coordination of many different pathways. So we make these proteins, they don't last forever, and we got to be able to control and react to the outside environment by either increasing the amount of that protein or decreasing the amount or activity of that protein, right? So proteostasis is the maintenance of like constant normal protein activity. And there's a lot of things that are involved in that. You have basic polypeptides formed from the ribosome. Will form a folding intermediate before it makes it to that native fold protein. And a lot of times, making the native fold of a protein is accomplished with the help of chaperones. Chaperones are proteins that will bind, oftentimes to a polypeptide as it's coming out of the ribosome to prevent. So a lot of times when it comes out of the ribosome, it's not the whole sequence yet, so you're going to have to protect some vulnerable sequences. So like you have ribosomes next to each other and all of a sudden spitting out hydrophobic regions, they can collapse together and form cross beta sheet structures and just precipitate out a solution as they're being made. You see that cross beta sheet structure is what you see with your amyloid proteins that are, like prion protein, amyloid and Alzheimer's, Huntington's disease. Those are all insoluble protein misfolding disorders because they form cross beta sheets between other proteins and are insoluble. So chaperones can help with that as they're being made to help them fold the native protein fold. This native protein fold is only going to have a certain half-life until it'll degrade chemically, and then it'll also be degraded by enzymes in the cell when you don't need that protein. So those turn into peptide fragments, and that's degradation. It can be helped by degradation pathways such as the ubiquitin proteasome system. What does ubiquitin mean? Or ubiquitous, right? So ubiquitin is a peptide that they discovered that they found it everywhere in all the cells, added to all kinds of different proteins. It's a peptide. So when a protein gets misfolded, when it gets old, it's marked for degradation. This ubiquitin proteasome system, there's a ubiquitin ligase is an enzyme, recognizes a misfolded protein. It'll add this ubiquitin tag to it, which is a mark to be ubiquitinated even more, so you get multiple copies of ubiquitin added to it, which is then a signal for this misfolded protein to be sent to this proteasome, which is like a big trash can. Ubiquitinated proteins go in and little fragments come out. So chaperones will remodel proteins, and they can remodel it back to the folding intermediate and help it to be refolded into a good protein again. Here's your aggregation again. So you have amorphous aggregates. If you have a folding intermediate, it forms cross beta sheets with the neighboring protein that'll aggregate. And this is amyloid fibrils. You see these beta sheets. You can have beta sheets between neighboring proteins and causes them to aggregate. And autophagy is where the cell will just digest proteins itself, not using the ubiquitin proteasome system. It's called autophagy. Now it's one fragment. peptides also. So this is your proteostasis, the maintenance of the proteasome or proteobiome in the cell, basically. So protein stability and folding. So protein's function depends on that three-dimensional structure. If you lose that integrity, it's accompanied by a loss of activity, and that's known as denaturation or unfolding. If you unfold that protein, it's not going to be able to be active anymore. We can denature proteins by adding heat or cold. If you put energy into that system, it's going to give it more entropy, allow more rotations around different bonds, and it's going to cause it to unfold. We're going to put energy into it, you can break hydrogen bonds, and it's going to unfold. In extreme cold, you might need something to be able to rotate. Well, oftentimes, you know how you make aliquots in the lab with proteins? You do that because when you heat and thaw proteins over and over again, they form those cross beta sheet structures and drop out of solution and go bad. So consistent thawing and freezing of proteins, like if you have an enzyme that you want to use over and over again, it's good to aliquot it in small amounts. So you take the aliquot out, use it, and then throw it away so you're not constantly freezing and thawing. So that going back and forth between freezing and thawing, you lose proteins every time. pH extremes are going to affect how the proteins fold, right? You get secondary structures done through the amide and the carbonyls. Those are both protonatable or able to donate that proton. So that can affect folding. And then you got, salt bridges between side groups. They're going to give you structure. Those are protonatable or deprotonatable groups that are going to be affected by pH extremes. So that's one way to unfold a protein is you alter the pH to an extreme. Organic solvents also, because globular proteins, those, hydrophobic domains collapse to the center of the globular protein. If you dissolve that globular protein in an organic solvent, it's going to unfold and, you get that big increase in entropy. And you don't have to worry about the entropy of the solvent because those hydrophobic domains are happy in the organic solvent, right? Chiotropic agents, and those are salts such as urea and guanidinium. You guys know the structure of urea or guanidinium? Guanidinium, if you have the amino acids there, it's the one, where is it? The arginine. At the end of this arginine, this nitrogen, and that carbon, that's a guanidinium. And it's just these two. It's guanidinium. And you can actually, that's one of the ways, like when I would work with brain tissue that had amyloid plaques, to dissolve those amyloid plaques. Use high concentration of guanidinium, or you can use urea also, and it interferes with those cross beta sheets because of all those nitrogens. It can hydrogen bond and break the cross beta sheet structures and unfold just basically any protein. So. And this shows you some data about like melting temperatures. So the melting temperature of DNA is when you separate those strands, or the temperature at which 50% of the strands are going to be separated. Same thing with the melting temperature of a protein, is at which you get 50% of the protein that you have in solution is going to be unfolded. So down and around physiological, you have a really low amount of that protein unfolded.
So it's denatured way before the melting point, and the melting point is just like...
Well, a portion of it, like at physiological temperature, is always going to be unfolded. There's an equilibrium, it goes from a folded state to an unfolded state, right? Back and forth. So as you increase the temperature, more of that, the melting temperature is when 50% of that protein in solution is going to be unfolded.
When it's unfolded, is it just out of its native state, or is it like totally unfolded?
That's a good question. Unfolded, the way we just define it, is just so it's not going to have structure, or not structure, function.
Okay.
So I suppose it's just when it doesn't have function. But you can totally unfold it eventually.
That's extreme.
Yeah. So this is two different proteins, and proteins, of course, are going to have different melting temperatures depending on their structure. Ribonuclease A versus apomyoglobin. Apomyoglobin is like a precursor to myoglobin. This is ribonuclease A again. This is with increasing concentrations of guanidinium, and you get nearly 100% at about 5 molar guanidinium, which happens to be what I would use to extract amyloid from precipitated amyloid from Alzheimer's-like brains. And one of the cool things is how the proteins, once you unfold them, they can spontaneously refold if you bring the conditions back to, that are conducive to have the native state. And what they did with ribonuclease is they purified ribonuclease, a small protein that contains 8 cysteines linked by via 4 disulfide bonds. And urea with 2 mercaptoethanol will fully denature the ribonuclease. Do you guys know what the 2 mercaptoethanol is?
Yeah.
Huh? Beta mercaptoethanol, right?
Yeah.
So that adds a sulfhydryl group. It allows for the reduction of the disulfide bonds, so it breaks those disulfide bonds and gives you the free thiols and the cysteines. So basically a way to take apart those disulfide bonds. And then, so when the 2-mercaptoethanol is removed and the urea, this ribonuclease A will spontaneously refold and reform those disulfide bonds in the correct locations, and then have a completely functional native conformation. So it's quite a simple experiment. It was also important and earned Chris Anna Finsen his chemistry Nobel Prize in 1972. And this is basically how it is. So in your native state, ribonuclease A, you add high concentration of urea and beta mercaptoethanol or 2-mercaptoethanol. The 2-mercaptoethanol is basically, we know the structure of ethanol, right? 2 carbons, 1 OH group. So this is carbon 1, but 2 just has an SH group. That's 2-mercaptoethanol or beta mercaptoethanol. So it's this SH group that's able to keep these cysteines reduced. Urea unfolds all the secondary structure, makes it, what's it called? Random coil type structure, right? Which is really unstructured, so calling it type of structure is redundant, right? Not redundant. I don't know. I can't think of the words I'm trying to say. You remove the urea and mercaptoethanol. Anybody know how you can remove the urea and mercaptoethanol?
Precipitating water.
Could do that, but the way they normally do it is totally different. So you remember the protein's kind of big versus urea isn't really much bigger than this. Huh? Straining, yeah, it's sort of straining. What's sort of like straining? No, you can't do that. You could do it that way, but it's way easier if you strain it this way. Dialysis.
Oh.
So you dialyze it away the urea and beta mercaptoethanol, and it'll spontaneously refold, and that'll have activity to ribonuclease, means it breaks down RNA molecules, takes them apart. And protein folding is going to follow a distinct path. So you're going to have these local minima in structure that are going to fold first. Like you have this structure here, and it's going to want to form these cross, anti-parallel beta sheets. This automatically is going to start spontaneously forming alpha helices and then more beta sheets. And then these will come together based on their R groups. The solvent's going to, want to force those hydrophobic R domains together. Charges are going to want to interact. It's going to spontaneously fold eventually. So how do they fold so fast? They're going to fold to the lowest energy fold in the microsecond to second time scales. So it's Microsecond is how many? It's like 10 to the minus 6 seconds. So it's like 1 million microseconds in a second, right?
Yeah.
Yeah. And it's mathematically impossible for the protein folding to occur by randomly trying each conformation. That's, I guess that could happen, but that it's impossible for it to fold that fast when it does that. So what it's going to do is it searches minima is not as random because the direction toward the native state structure is thermodynamically most favorable. So as it folds to that native state, there are little regions like we just showed that are going to want to come together first, and it's going to kind of encourage the other folds to happen as it starts to come together thermodynamically. And you can end up with, this is like diagrams of protein folding, like versus delta G, like changes in free energy. The lowest state, this is going to be the native fold. You might find systems where you have a lot of localized maxima and minima. Cases like this, you might need chaperones because you could get trapped in a trough or something without chaperones. But more often they're like this. Ribonuclease A might be something like this because it does it so well by itself. Not so often things like this. Here's you can be trapped in a trough there also. Makes it all the way to the localized. Hence, I was talking about chaperones. They prevent protein misfolding, and they help take a folding intermediate and form a native protein. A lot of chaperones, the first ones that were discovered, they're often you'll see chaperones are called heat shock proteins, HSP, because they were discovered when they were heat shock cells and see what proteins were being made after they heat shock different cells. And a lot of cells upregulate the production of heat shock proteins. This one in particular is heat shock protein 70, which has, according to this, 2 different affinities for ATP versus for ADP. And it'll bind these folding intermediates, burn that phosphate, and help make that native fold. And it has a high affinity for it. So that's why they're called heat shock proteins, because they discovered that a lot of heat shock proteins are also chaperones. There's other chaperones also. There's these large complexes too that we find in bacteria and in eukaryotes called the chaperonins. The GRO-ES complex is one where you have a folding intermediates delivered to this complex, goes inside, and then each time the folding intermediate makes contact with the wall of the chaperonin, uses up an ATP, and you get a little bit more of a fold and a fold. You get multiple ATP hydrolysis before eventually it gets in, and then as it comes out here, we have the native fold that comes out of the complex. It's an ATP-dependent process. There are certain proteins that'll bind to it like hundreds of times before it comes out as a folded protein. So it takes like several hundred ATPs to fold. And this is from the Dicker book showing you that complex. And then we talked about cross-baited sheet, amyloid fibrils, and whatnot. And this is what we find. It's a protein misfolding is the basis for numerous diseases such as Alzheimer's, Huntington's, Parkinson's. They're all protein misfolding disorders where you get a misfolded, prion disease is also partially misfolded, which can spread and cause other normally folded proteins to form that same misfolded. They form these cross beta sheet structures that ultimately fall out of solution and form these amyloid fibrils that cause it to be, they form protofilaments and then larger amyloid filaments. Drop out of solution, aggregate outside the cell or inside the cell, depending on the disease and the protein.